Hi, my name is Tomasz Grzegorzek, and it's my pleasure to present our work on a deep generative model for AMP discovery called HydrAMP. The development of resistance to conventional antibiotics in pathogenic bacteria poses global health hazard. It is predicted that by 2050, antimicrobial resistance will lead to 10 million people dying every year, and that's more than all types of cancer combined. Antimicrobial peptides could be a partial solution to this problem. It has been shown that the bacterial resistance against AMPs would evolve slower than against conventional antibiotics. The discovery of new AMPs is challenging. Combinatorial space of peptides is huge. Moreover, not every sequence is valid. Some peptides cannot be synthesized, and few manifest antimicrobial activity. The high cost of wet lab experiments results in a limited number of experimentally validated peptides. AMPs are represented as sequences of amino acids. Novel AMPs are usually discovered by trial and error process of substituting and/or introducing certain amino acids into already known AMPs. These small changes can substantially affect antimicrobial activity. Therefore, we need a non-sparse representation of peptides that must also encode antimicrobial properties. We would like to have a method that can suggest meaningful improvements in the existing peptides. We would also like to generate AMPs de novo. A perfect candidate for AMP has high probability of being antimicrobial and high probability of having low minimal inhibitory concentration, MIC. Additionally, we know that most AMPs are cationic, which is described by high charge and high isoelectric point. These properties should be preserved in the generation process. To fulfill these goals, we have developed HydrAMP, a method based on conditional variational autoencoder that provides a latent representation of peptides. Each peptide representation consists of two parts, style coding Z and condition coding C. Each element of the style coding follows the standard normal distribution. The condition coding C consists of probability of being antimicrobial and probability of having low MIC. The representation is generated by variational encoder. This basic setup corresponds to reconstruction training mode. We added two pre-trained classifiers, AMP classifier and MIC classifier, that provide conditional coding and confirm antibacterial properties of generated peptides. We use the feedback from the classifiers to improve the coding. We introduce an additional loss term that requires the target condition C and predicted condition C' to be the same. To imitate analog discovery, we introduce prototype training mode. We use an existing peptide as a template to create a more active analog. Generation de novo is emulated in the unconstrained mode. We sample a random vector from the latent prior, and we force the model to use it to generate a novel peptide with set properties, either an active or inactive peptide. Reconstruction, prototype, and unconstrained modes are trained at the same time. The three modes of generation correspond to three heads of Hydra, hence the name HydrAMP. To avoid encoding antimicrobial properties within style coding, we impose the disentanglement of the condition coding C' of the generated peptide and the style coding Z through gradient regularization. That is, we demand the gradient of C' with respect to Z to be equal to 0. This is referred to as Jacobian disentanglement regularization. Additionally, the latent regularization term ensures that the decoded peptide is encoded by the same mu as input peptide. Now let me show you how HydrAMP performs in generation. First, we show that our model can generate novel peptides with requested properties. When prompted to generate active peptides, our model generated peptides with high probability of AMP, and the opposite in case of inactive candidates. In case of HydrAMP in the positive mode, the distribution of probability of AMP is skewed to high values. Other models are unable to generate active peptides with such success rate. We compared how well our model performs in the improvement of existing peptides with other models. We aim to increase the probability of AMP and having low MIC with respect to the input peptide. On panel A, we show improvement of positives, that is known AMPs. On panel B, improvements of negatives, peptides lacking antimicrobial activity. In both cases, our model is able to improve more peptides than other models. Our model can convert the peptide without antimicrobial activity into peptide resembling AMPs in terms of isoelectric point and charge. Although for low temperature this improvement is small, larger temperature inflicting more changes in the amino acid sequences leads to analogues resembling known AMPs. We selected a positive and a negative analog of pexiganon for further validation. The molecular dynamics simulation confirmed that the positive analog named HydrAMP was able to penetrate a bacterial membrane. The negative analog is unable to obtain desired conformation and remains outside of the cell. Wet lab validation proved that the positive analog is more active than pexiganon, while the negative analog lacks antimicrobial activity. To make our model accessible to a broader community, we provide a web service with all functionalities of our model. I would like to thank my supervisor Ewa Szczurek, my colleagues from University of Warsaw, and my collaborators from the Center of New Technologies and Medical University of Gdańsk. Thank you for listening.